Number one, we have a very unique immunotherapy platform. Immunotherapy is the most advanced and the most utilized therapeutic modality today in cancer. And it's a very, very crowded field because the current drugs, such as the checkpoint inhibitors, PD-1 and PD-L1, are in the over $20 billion in value and growing rapidly. And therefore, everyone in the cancer field has joined the bandwagon. And so it's unusual for a company such as ours to have a distinct platform that has advantages, at least perceived advantages over other technologies. We have seen some promising clinical activity to date. It's very early, but very promising in that it works in cases where you wouldn't expect to see activity with checkpoint inhibitors alone. And we, as I will explain to you in a few moments, we are addressing markets that very few, if any, other companies are addressing. And I think that's important. If you realize today there are thousands of clinical trials out there in combination with checkpoint inhibitors. So even if you have a positive result, you kind of get lost in all of the noise that's out there. And what we have done is we've gone to very unique patient populations and markets that are also very large. And so if we're successful, we believe that we will be an attractive IPO candidate by 2023. Before I get to the story of NeoTX and how we got to where we are today, the most important thing is it always starts with a good team. And you have just met our Chief Scientific Officer, Roger Kornberg, who, as you can see, is involved in many other programs. But his dedication to NeoTX is very, very high. In the audience, pleased to show, we have here the man with the mustache, Dr. Marcel Rosenzweig. Marcel, if you look him up on Google, you'll find that he was the godfather of chemotherapy. He was 20 years at the BMS, and against every one of his superiors, including the CEO of BMS, went on to develop blockbusters in cancer. When at the time when he joined, cancer therapy was pretty much a hobby for BMS. Also on the team is Ramona Lloyd, who has more than a dozen drugs that she brought to registration and approval with the FDA. She's the head of our regulatory side. Also, Michal Shachar in Israel, one of the best R&D, heads of R&D that there are in the country. Robert Harrow is a recovering investment banker, and his major component is he watches every single dollar being spent by NeoTX. And finally, Rob Kramer, who's actively involved in the company, was head of discovery at BMS and also at Janssen Pharmaceuticals. So what did we do? We came out with a kind of an original idea. You know, Google was probably the first to adopt this idea, which is the importance of information. weather the information. And I think also one of our board members, Tom Hill, who now is at 2 Sigma, has also the same philosophy. Nothing is more important than information. And in drug development, it is also true. And so we were thinking, where is the most information is in late clinical trials. And if the trial is successful, well, then we have no chance for a small company to in-license it. But if it was unsuccessful, you have this very interesting dynamic in that while you have lots and lots of data, the perceived value is low. So, if, you know, everyone out there always wants to find a bargain. These are bargains. And we use the vast experience of our team to find the right drugs that have failed in clinical trials and have the most data. Now you might think, okay, well, if a drug failed, isn't that negative data? And I think this slide here kind of represents how negative data can be positive. You see the black negative data of the 2 faces readily define the goblet in the middle. So the boundaries of negative information give you the positive information that help us to develop future clinical trials after a drug has failed. And if you look at today's cancer immunotherapy, probably the most important economically and medically today are the checkpoint inhibitors, which have been approved for several years and are the first-line choice for many cancer indications, and CAR T, which has been shown to be probably one of the most effective drugs of all time for certain blood-based tumors, but has of yet have not been shown to be effective for solid tumors. And we're going to focus on those 2 because we think that our technology fits kind of like a hand into a glove for these 2 approaches. Now, this slide just gives you a basic idea of what a checkpoint inhibitor is. As you can see here on the left, the interaction between PD-1 and PD-L1 between an immune cell and the cancer causes a stop signal and prevents the immune cell from killing the cancer. If you prevent that pathway from occurring, that interaction occurring, such as done with checkpoint inhibitors, as you can see on the right, our immune systems are perfectly adapted for killing cancer cells. And I'm not going to go into all the things that could go wrong, but you can see here in this slide that there are many, many causes of treatment failure for these checkpoint inhibitors. And you can imagine that that'd be true because the immune system is one of the most complex systems in the universe. And there are many components to it. And therefore, many things can and will go wrong. And the tumor is actively trying to survive. And so these failures of the immune system cause checkpoint inhibitors to be ineffective for the majority of patients. On the CAR T side, the idea behind it is to take the immune cells out of the patient. They are then genetically engineered, and then they are reintroduced into the patient with the genetic engineering that causes them to be to target the tumor. And as I mentioned, while this approach has been shown to be very effective in blood-based tumors, it has shown no efficacy in all the solid tumors. However, it has attracted the attention of major drug companies, and I read a recent article stating that there is over 108 companies out there that have some CAR T approach. And again, many things can go wrong that prevent it from working in solid tumors. And in addition to everything on the previous slide that can go wrong with the checkpoint inhibitors, here you also have an issue with the activity of the CAR T cell itself, which is often exhausted by the time it gets back into the body. And so the way to think of it is our immune system is a very, very leaky pail of water trying to put out the fire of cancer. And almost all approaches in immunotherapy, they deal with a particular hole or several holes of the pail, but cannot really stop all resistance mechanisms from occurring. So if you plug it up in one place, it's just going to spill out somewhere else. And let me see, I hope this works. We have a little cartoon here to show you how our drug differs from that and how our drug works in creating a holistic approach and a de novo immune response against the cancer that's directed against a bacterial target. Uh-oh. Say, oops. Could you make the volume higher? I can't go. Is that possible? If not, we'll just have to listen in carefully. Okay?
NeoTX's tumor-targeted superantigen platform, or TTS, offers new hope for patients with advanced solid tumors. The TTS molecule has two components: a Fab against tumor-associated antigen, a molecule that attaches to cancer cells, and a superantigen peptide, which is a bacterially derived peptide that can activate a controlled, natural, antibacterial immune response. Once injected, TTS molecules circulate through the bloodstream and enter the lymphoid organs and the tumor. In the spleen and other lymphoid organs, TTS molecules bind only to specific T cells and to antigen-presenting cells. This interaction results in activation and proliferation of the specific T cells. Typically, those superantigen reactive T cells represent less than about 2 to 3% of the total cells at baseline. After the administration of TTS, however, they can increase to around 30 to 40% of the total T cell population. The activated T cells prime to recognize and attack the superantigen traffic to the tumor. At the tumor site, TTS molecules bind through the Fab part to the tumor-associated antigens, coating the tumor with the bacterial superantigen. Superantigen reactive T cells that traffic to the tumor recognize the superantigen, bind to it, and attack the tumor cells by releasing granzyme B and cytokines. The antigen-positive tumor cells are killed by granzyme B, and the surrounding antigen-negative tumor cells by cytokines, TNF-alpha and IFN-gamma. This dual anti-cancer effect continues until the tumor is eliminated.
So the point of it is not to deal with a specific feature of the immune system response, but to create a brand new one. So we don't fix a broken immune response, we replace it completely with the antibacterial immune response. So to show you some of the clinical trials that we've done in the past, and to give you a little taste of what we are doing currently, and then finally some of the plans for future clinical trials of our lead compound, which is hard to pronounce, neptumumab, and we call lovingly NAP. So here's the highlights from the previous trials. This is everything we have to date. This is from our licensor and our own trial results. First, in monotherapy, where we've shown some high survival. Secondly, in combination with chemotherapy, where we had some exceptional responses. In the phase 2 trial, in combination with interferon alpha, is where we found that's where the trial had failed. And we believe is because interferon alpha increased the anti-drug antibody responses, and therefore these high anti-drug antibodies interfered with the drug. And to show that and to convince ourselves that that's true, we did a statistical analysis of the small group of patients that did not have high anti-drug antibodies, and we showed that they had a severe survival benefit in that subset. And then finally, in our current clinical trials, we've had a complete response and several partial responses, and we'll be talking about that in a few moments. First, the phase 1 as both in a single agent and in combination with chemotherapy. In both trials, we showed good safety, and we also saw encouraging data of survival. In the monotherapy trial, the gold standard, and really if you have cancer, the only thing you really care about is how long you're going to live. And in this case, we looked at the survival after 2 years, and we saw in non-small cell lung cancer, and we saw a really exceptional survival rates of 42%, which compared very favorably with the survival benefit in checkpoint inhibitors, which is slightly more than just half of that amount. In combination with docetaxel, chemotherapy, there were 2 patients who had a partial response, and one of them, here you can see on the left, the patient has no immune cells or T cells that are in the associated with the tumor. And if you have no T cells, then no immunotherapy is going to work because that's what you need. And so you could see that that after therapy, there's a massive infiltration of T cells into the tumor, and that's what we call making a cold tumor hot. And when a tumor is hot, the checkpoint inhibitors are known to be highly effective. So we serve the cold tumor, make it hot, and then the checkpoint inhibitors could then kill the tumor. One of the cases we saw in this trial was really exceptional. Was a patient who had stage 4 lung cancer that was metastatic to the liver, and who had a very, very short life expectancy, was given this combination. This also a patient who was already resistant to chemotherapy before they were ever given this drug. And it was really a Hail Mary. The patient was on therapy for 6 months and then managed to live 11 years without any additional therapy. And that just shows you that our drug could reinitiate the immune response and keep it going even after the drug is no longer administered. We have a current trial we're in collaboration with AstraZeneca and their checkpoint inhibitor. It's shown acceptable safety. There are 3 patients who have stable disease, and we have also 3 patients with objective responses, And these are all in patients that are really rare to show response to immunotherapy. We had calculated that the probability of getting these 3 objective responses alone with just the checkpoint inhibitors and not our drug is in the neighborhood of about 1 in 10,000. We have started a phase 2 trial in the US. These are for patients who already failed immunotherapy, and they will be given our drug in combination with chemotherapy. These trials are just starting to enroll now, so of course we don't have yet results. On the planned trial side, what we're planning to do is one is a planned trial with checkpoint inhibitors. And here we're going to use precision medicine, but we're not going to use it in a traditional context. Normally with precision medicine, you look for patients who you know are going to respond to the drug through molecular screening. So depicted here in green, as opposed to patients, let's just say the red patient would be one who is highly unlikely to respond to drug. So you select for what you want, which are patients to respond to drug. What we're going to do is we're going to select for patients who are known not to respond to checkpoint inhibitors through biomarker selection, and use this patient population to give our drug in combination with the checkpoints. If we see activity here, it's a foregone conclusion that it's because of the activity of our drug, since we know that these are patients who do not respond normally to checkpoints. And this is a very large market, and to the best of our knowledge, we are the only company that is currently selecting for this patient population. On the CAR T side, whatever our drug can do to natural T cells, it can also do to these engineered CAR Ts. And there are essentially 2 places of entry that our drug could be helpful. One would be in the manufacturing step while they are genetically engineering the T cells from the patient, and 2, in the treatment modality, to add to patients. And in this way, we think we can make CAR T effective in solid tumors. This is just a in vivo result that was done not by us, but this is done by an independent lab and was published in PNAS, where they used the bacterial component of our drug and showed that they can make CAR T effective in solid tumors. And in the case of this published report, 4 out of 6 mice who were treated with our with superantigen had a complete response, and their tumors had disappeared, as opposed to 0 for those mice that did not have the superantigen administration. The market potential for what we're doing, if you know anything about this business, is huge. It's in the kind of wrong place, but essentially the market for the precision medicine is over $8.5 billion worldwide for peak sales. That's for our biomarker-derived trial that I explained. And in the CAR T, it is also somewhere around $8 billion in market potential. That combined with our second-line lung cancer trial gives us a total of over $27 billion in peak sales of market potential here. So very huge markets, yet we're going after markets that are not really currently being addressed by most other immunotherapy companies. We're trying to raise now 85 million. We are also, our lead investor is Larry Ellison, but we welcome other investors to come aboard and join us in our journey. And then I'll just conclude with this slide because it kind of explains what our drug does in case you hadn't been focused until now. The tumor escapes immune attack by being invisible. And our drug coats the tumor with a bacterial target to make it visible and then attackable by the immune system. Thank you very much. Appreciate it. Okay?
Yes, question.
I'm sure that was very impressive. I'm sure guys are just looking to throw money at you. What are you looking for in an investor?
We would prefer ones that have money. As a rule, there's a reason why we're at this conference today, and I'll explain to you why. Be very transparent with everybody.
I appreciate that.
The traditional venture funds and crossover funds have not shown a lot of enthusiasm in our program. And the reason why is because they don't, there's probably several reasons, but the major reason is they don't agree with our initial thesis, which is to take a drug that has failed. They believe that those drugs, if it failed, it's, the industry does not appreciate its value. So as a starting point, we have had less interest from traditional funds, and that's why we went to sophisticated investors like Larry Ellison, who did a very good job in vetting us, and others like Tom Hill is in this room, but I would say that the venture funds and crossover funds will come on. Even they will, I think, eventually agree, but at a point where we have more data. So we have promising data, but everyone knows, if you know biotech at all, you know that promising data doesn't always translate into approvable drugs. The fact that we can go very rapidly and for relatively low amounts of money in these markets that I've addressed, I think makes it a very attractive investment, but probably we will not be going for the traditional funds, but we welcome funds that, from, as I say, people who do have money and also have enthusiasm. I got to tell you that I spent my whole life as a serial entrepreneur, and at times have been funded by traditional venture funds and untraditional funds. And as a rule, I would say it's been much more fun to be involved with the non-traditional funds, and not less helpful either. Many traditional funds, I think, have a problem that they don't always know what they don't know. And that's a dangerous person when they're in charge, when they're on your board or directing the direction of a company. I like our present board composition because they're highly intelligent, highly successful, know what they know, help me. No one knows everything. I know, this is a very complex business, and it can be helpful, but also know what they don't know and leave the actual drug development to experts like Marcel and others to run the company effectively. Does that answer your question? It was
Yes,
very long.
It does.
One.
Thank you very much,
Okay.
sir.
Anyone else?
Thanks, Asher. You mentioned you were seeing anti-drug antibodies earlier in your studies. How have you addressed that issue, and what's it currently look like for those?
So we saw some issues with anti-drug antibodies in the current study as well. Some patients had very high anti-drug antibodies, as you would expect from a drug that has a bacterial nature. Our bodies are highly attuned. For the same reason why the drug works, you would have anti-drug antibodies. Our bodies are very attuned, immune systems are very attuned to a bacterial infection, and it causes a strong immune response. We are currently administering as pretreatment a drug called obinutuzumab that prevents anti-drug antibody. And we have seen very encouraging signs that for at least the first few cycles of drug, that these anti-drug antibodies have been remained at bay. So we're exploring both obinutuzumab and other potential drug combinations that will help lower the anti-drug antibody and create even stronger and better responses.
And your CAR T program looks really interesting. Have you had any reaction from the KOL, key opinion leaders in that area? And can you give us a little color on that?
Yeah, my experience in life has been when you go to known opinion leaders in the field, almost always I get a response. One usually loves it, and they're like, this is the best thing ever. One hates it, and there's like maybe 2 or 3, if you go to 5, that are sort of in the middle. That's almost always happens. In our case, we've gone now to, I would say, 3 KOLs, maybe 4, if you include Mariel Schnall, who's the past president of SITC, the Society for Immunotherapy, but not an expert per se in CAR T. But all of them not only have been interested in and we're excited about the program, but all of them want to collaborate with us. So we already have a collaboration with the first guy we talked to. We're in the process of having it with the second, and hopefully we'll have all 3 of them collaborating with us. I think the opportunity in CAR T is exceptional. There are other therapies that are being contemplated to increase the efficacy of CAR T. As you might imagine, this is a field that has huge market potential, and it's already, it seems like so maddeningly close. It works so well in some blood-based cancers and works so poorly in solid tumors. Yet I think our drug, for reasons that I sort of spoke to quickly here, but I can get into much greater detail, our drug is perfect for CAR T, and could, I think, better than almost any other, because it gives us holistic attack, is going to be quite effective in the CAR T space. And I think we can, by next year, we can start clinical trials with CAR T. You and with the ugly...
At least I have a new haircut.
Yeah. It's that beard.
It's the beard.
Yeah.
So what's the takeaway in terms of the durability of response? Does the holistic aspect of DAP give a chance of having much more durable response for both the checkpoint combination and CAR T?
Yeah, that's a great question. I guess I'll pay the $10 afterwards for asking it.
I just wanted to make sure you got across the essential point.
Yeah. Creating a bacterial immune response against a tumor reinitiates the entire immune response. And when you have that, when everything is lined up and all systems are green, what you have is an immune system that potentially, and I say potentially, can wake up and say, hey, there's a tumor over there and I'm going to kill it, independent of any drug. And that's really what you want, because every drug, as you know, every drug, and any drug that will ever be designed, in my opinion, will always be subject to resistance. And in the end of the day, all drugs will stop working against the tumor. Our drug, because it is a natural initiation of the immune response, might cause, and was seen to cause in animal models at least, in every instance. Once we were able to, for mice that became, that their tumors were complete responders, so they had no signs of cancer anymore, we tried to re-inject tumor cells back into these mice to see if we could cause the cancer to reappear. And in no cases have we ever found that the cancer reappears, meaning that the immune system. What?
You out-cancered the cancer.
Right. I mean, if there was a big market for cancer drugs for mice, we would be billionaires today. It really works well in mice, and we think that one patient who lived 11 years might also be an early indication of this. It's called epitope spreading. This phenomenon of reinitiation of immune response, a natural immune response against the tumor.
Thank you, Asher. I'll tell you, I've known Asher for about 6 or 7 years now, and he's definitely in the habit of under-promising, over-delivering in very mellow tones. So I really enjoyed your presentation. Let's take a quick break. We're slightly behind on time. Going to be back in just a few minutes to hear from Adam Wolf.